Good morning, everyone. Friends, today we are going to discuss about the most important topic related to chemistry, that is analytical chemistry. Analytical chemistry is about the exploring of science and identifying, measuring and understanding the chemical substances. It is a branch of chemistry which involves study of chemical analysis which focus on identifying the substance, describing their properties and determining the composition, percentage and amount of substance present in that particular compound. Since everything we use in day-to-day life is contain chemical, and understanding the composition of that chemical substance is very necessary in your day-to-day life. That's why we will focus on this topic. This field encompasses the identification, measurement. It also involves the structure determination, purification and separation techniques. We have to discuss all about these points in analytical chemistry. And our today's topic is basically a introductory idea about analytical chemistry. As I have already told you that analytical chemistry is a branch of chemistry which involves identification of substance, separation techniques, determination of quantity of the substance. So is it is involves in all branches of science like life science, earth science, applied science, biology, ecology, medicine, and each and every field where we have to measure something and where we have to identify something. That is, analytical chemistry bridges multiple scientific fields, drawing from and contributing the diverse area of research and application. It is also related to core chemistry branches, which utilizes the principle of physical, inorganic, and organic chemistry plus chemical engineering methods. It is also involves in essential progress in biology, biochemistry, geology, geophysics. Because today there is so many researches going on in physics, geology, and biochemistry, which involves analytical chemistry. So it indicates that the analytical chemistry subject is having an interdisciplinary nature because it connects various fields and it is widely used in medicine, industrial, various government departments, analysis departments like forensic science and academic laboratories in worldwide. The scope of analytical chemistry is huge. It has a remarkable wide encompassing manual, chemical and instrumental method across numerous applications. Here alone, we can't predict that whatever maybe the analysis methods are going on in your laboratory because there are so many sophisticated laboratories are present all over the world, which which work there on an analytical chemistry. Like agriculture, here we have to test nitrogen, phosphorus, potassium levels in fertilizer. In case of food safety, which is most important point for the point of your day-to-day life, that is measuring the protein, vitamins, carbohydrate, fats, contaminant in food and beverages. Medicine, whether the composition of medicine which we have taken is correct or not. Again, forensics department, manufacturing of some product to determine the quality of that product, analytical chemistry make a pivotal role in that all these branches. Basically, there are two types of analysis, chemical analysis. One must is qualitative analysis and another is quantitative analysis. As the name involves, quantitative means it measures how much amount of composition present in the given substance. That can be determined by volumetric, gravimetric or various instrumental methods like titration which perform in your laboratory, gravimetric method, estimation, that's experiment, that is the estimation of nickel by nickel DMG. This is about quantitative analysis. That is, here we find out the quantity of the substance. While in case of qualitative analysis, as here it indicates that two test tube, in one test tube we have added drop by drop another solution, and here the some precipitate is occurs in the lower portion of the test tube. That is, this is a quality. In this qualitative analysis, we have to find out the whatever may be the cations or anions of the substance uh, present in the substance, unknown substance. That is, it determines what may be the constituent present in the given sample. It can't determine the amount because it is determined in quantitative analysis. Therefore, in qualitative analysis, we can only identify which ions or which element present in the given sample. That is determination of constituent. Now, for this quantitative and qualitative analysis and for analytical chemistry purpose, first we have to collect the sample. It is the foundation of analysis. There are so many methods of sampling. That is, it is a process of extracting a small portion from the bulk material such that the sample truly represent the composition as a whole. That's why proper sampling is very important in analysis process. We can't check all the substance because that substance may be present in various branches in large amount. So we can't analyze that whole substance as a bulk material. So we have to take some sample. So there are actually basically four methods of sampling. First one is increment. That is, it involves small portion of the sample taken from non-homogeneous bulk material at different locations. Means we have to take the sample from different locations. Another one is gross sample, which involves combined mixture of all increment, generally too large for direct analysis. And third one is subsample. It is the random it is the reduced quantity obtained by dividing the gross sample. That is, whatever may be the gross sample we prepare from that increment, that must be reduced to some particular quantity by dividing the gross sample. It is subsample. And another one is analytical sample, final test sample after particle size reduction and through mixing. One more is that is random sampling. It is non-systematic selection from a part of homogeneous bulk material. Means here we have to collect the sample from any part of the material, that bulk material from we can take, there is no systematic way to collect the sample. That's why it is known as random sampling. And in systematic sampling, scientific selection at a fixed interval uh, is taken in systematic sampling. Now, accuracy and precision is another important point in analytical chemistry for the analysis point of view. So, accuracy as itself indicate that accurate results must be obtained. Here the diagram itself indicate that all these four cross are at the center, at this yellow center, indicate that this is accurate result. Means all the values obtained are in close relation. But therefore, accuracy is determined as how close a measured value to the true or the most probable value compares to the measured result against a known standard. It is very well explained by the example. If a true value is 25.0 and after analysis we have get the result of 24.9 or 25.1, that it is show high accuracy, means your result is mostly accurate. While when we discuss about the precision, it is how close repeated measurement are to each other under identical condition, shows reproducibility, not correctness. It is not a correct result, but it is reproducible. It can be very well explained from this example, that is result of 24.0, 24.1 and 24.0 ml shows high precision. Means here it shows that here it is not at the center. This cross, these four cross are not at the center. Therefore, it is not accurate but precise. Means that uh, four center must be in between this yellow circle, oval shaped circle. That four cross must be present in this yellow oval shape. But that four cross are present on this blue circle, that is outside the yellow, means these results are not accurate but it are closely related. That's why it is known as not accurate but precise. Here that four crosses are one is on the red circle, one is on the blue circle, that one is again on the very close to black circle. That is these results, whatever the results obtained after such a type of analysis, that are neither not accurate or not precise. It is not precise. But here, in case of this diagram, all these four cross are closely in between this yellow oval shape. That is, that results are accurate but not precise. Means these four cross are not very close to each other. Therefore, these are not precise. That is, high precision does not guarantee about the accuracy. An analyst may obtain highly precise value that are systematically far from the true value. No. Another most important point is what is the significant figures. So significant figures are the digit. It is a digit that indicates the measured precision of result. That is, the number of digit used should be matched to the precision of the measuring instrument. Now digits, when we discuss about the digits, so first we gone through the non-zero digit. What is non-zero digit? So non-zero digit is always significant. For example, 5.406 has four significant figures, while 0 between non-zero digit, the example of this is all these are always significant. For example, 5.406, the 0 is significant because here 0 has some value. 0 between non-zero digit, that 0 is present between two non-zero digits and it has some value. Therefore, it is always significant. Here, therefore, again I have told you that about non-significant figures is that significant figures are digit that indicate the measured precision of results. The number of digit used should match the precision of measuring instrument. Now, what is about leading zeros? That leading zeros are not significant. It can be very well explained from this example, that is 0.00405 has only three significant figures and trailing zero with decimal, that is, for example, 400.0 has four significant figures. In last rounding off value, what is about rounding off value? If last digit, whenever we discuss about any value or when we will write any uh, digit which include point, decimal point, that is if the last digit is greater than 5, increase proceeding digit by 1. That is, for example, 8.946. So it can be round up to 8.95. That is, this last digit is 6, that is above 5. That's why it's maybe rounding off to 8.95. This is about rounding off rule. So this is most important point. That is, when the digit is more than 5 and less than 5, what we have to taken. This is about that. For example, if the last digit is less than 5, drop the last digit. If for example, 8.943, so here 3 is less than 5, so we can drop it while writing on that somewhere paper in or in report. So we can drop this last figure if it is less than 5, and if the last digit is exactly 5, then it can be round to nearest even number. Now, in chemical analysis, whether it may be qualitative or quantitative, there are some errors are occurs during the analysis, and that error may be due to different reasons. Here, while doing practical volumetric analysis, here we have shown the burette and that reading. So how that burette reading is observed, this green line, this green arrow indicates the correct way how to measure the burette reading, while this red arrow indicates the non-correct way of observing the burette reading. So please remember this point, or remember this diagram, that is which we perform in laboratory mostly. So when we discuss about the errors, as I have already told you that there are so many errors, we can do the analysis on instrument, some methods, we can use some methods. Our personal error is also introduced. So there are so many types of error, that is instrumental error, method error, operational error, personal error, and indeterminate, which is known as random error. So how instrumental error arises? So faulty or uncalibrated instrument, or unbalanced scale, or improperly calibrated glassware. So while doing analysis, first we have to calibrate each and every instrument and glassware. It is very important for analytical point of view. Another is method error, premature indicator color change, unwanted co-precipitation, incomplete reactions, or interfering side reactions. Means whatever may be the analytical method which adopt for the analysis, we have to you must know about each and every reaction involved in that method, because incomplete reaction may affect our analysis. Therefore, before to start that analysis, you must be very well familiar with the reaction which is going on in that method, so that we can minimize this method error. Another one error is operational error. What is about operational error? So it is unclean apparatus, improper transfer, incomplete drying, wrong reagent quantities, or weighing hot sample. So as I have already told you that we have to adopt the exact procedure step by step while doing analysis. So first of all, apparatus plays a pivotal role in chemical analysis. You must clean the apparatus and dry it before each and every experiment, not only in case of analysis. So for each and every chemical experiment, first our first aim is to clean and dry the apparatus. And suppose if we have to transfer some amount of, for example, 5 ml of some quantity is we have to transfer in the solution. So we have to transfer exactly 5, not 4.8, 5.1 like this. And some process involves drying, cooling. So that must be accurate. While weighing the substance, your quantity must be correct. Another one error is personal error. That is, sometime some persons have color blindness. They can't determine exact color, or they cannot identify the color. That is inability to see color change. Sometime while doing the calculation, we have make some minor mistakes, lack of attention, carelessness during the procedure. So that is, you must be very prompt while doing your experiment. That is, sincerity must be necessary while doing chemical analysis. Another one error is indeterminate or random error. This occur by chance with no indefinable cause. They cannot be controlled or corrected, arising from uncontrolled external conditions or hidden factors in complex procedure. That is, if the procedure is very complex, it involves several steps. So we can't control sometimes the conditions that may be essential for that experimental point of view. So this is that type of error, that is indeterminate. It is, आपण त्याला मराठीत असं म्हणतो अनिश्चित, तशा स्वरूपाची ही एरर असते. Now once the error is arises, as we are doing some experiment, so error must be occur. So how to minimize that error? So it is a best practice while doing the experiment. That is, accurate analytical result requires systematic attention to detail and proper technique throughout every stage of analysis. So in order to minimize the error, our first and important point is that calibrate all the instrument and glassware. As I have already told you in previous slide, that ensure that all the burette, pipette which we have used in volumetric analysis must be calibrated before the starting the experiment. Whatever may be the procedure given in books or in that procedure, that should be carefully adhere to established protocol and avoid sample or product loss during the process. Most important point is select appropriate method. That is, choose the analytical technique that minimize error and use only pure and highly quality reagents. This factor is again important because without quality or without using quality reagents, our results are wrong. So use standard reagents for analytical purpose. Uh, uh, so our best practice is that purify the compound before doing the experiment. While doing in laboratory, you must be work with discipline and attention. That is, control all the experimental condition including pH, temperature and other conditions at throughout the experiment. If possible, use the buffer solution to control the pH. If possible, you use thermostat for controlling the temperature. And another one minimizing of error point is perform multiple verification. That is, repeatedly conduct that analysis. Conduct repeat analysis using different techniques when possible. We can't do the experiment by only single method. So we can do that experiment, and that is that analysis using different technique as far as possible. Thank you.